All right, so welcome everybody to our first GKS open house. It's a real privilege to be able to talk to you about the work stream today. I'm joined, as Beatrice said, by my colleagues Melissa and Larry. The three of us are going to be working through the agenda that Beatrice laid out for you, and I'm not going to spend any more time on that. We're just going to dive right in. So, thank you all for being here. Welcome to GKS. A little bit about who we are and what we do. So, we're very concerned with the challenges associated with the what we call the variant interpretation bottleneck. This process by which our frustrated clinical scientists need to pull together information from a variety of different resources in order to interpret the role of a variant in a patient context. And this involves looking through a number of different resources that describe information using various different standards or no standards at all for describing the evidence associated with a genomic variant. And they have to pull it all together. They have to piece together their support and ultimately use that to drive interpretation. It's very laborious. It's very time consuming, and it's a very costly way of doing interpretation. We're trying to advance that through the development of standardized reporting or standardized structures for sharing these evidence and linking them together into knowledge statements so that can then be reused by other groups around the world. So in order for us to do that, we require this coordinated development of genomic knowledge standards, which is what we do in the GKS workstream across a federated variant evidence ecosystem. And some of the challenges we see in in this ecosystem is that variant evidence is very disparate in its structure, it's very fragmented in its context, it's distributed across a variety of different locales. And so our solution must be interoperable across those data structures. It must be self-descriptive for reference context, and it needs to be globally consistent across this federated data ecosystem. And so this is kind of the way that we think about developing these standards to work across these different institutes. And of course, the challenge is only going to continue to grow as we see new forms of reference data sets in different builds with different technologies. Of course, we've seen, the telomere to telomere work. There are, population specific reference genomes. There are, functional studies that are high throughput that are on custom sequences. We need to be able to link all this together and use this across a federated network where people don't have, a single shared reference space. And in order to accommodate this, we also need to be able to describe, highly diverse statements about knowledge of variants, right? So here are some examples of different clinical knowledge, genomic knowledge statements that exist out in the world today, right? So here, for example, is a ACMG guidelines for clinical laboratory statement regarding Huntington's disease and its role with CAG repeats being above 40 or more in the patient. There's experimental variant data from a MAVE experiment here, where it scores as loss of function, supporting pathogenicity of a mapped variant to SCN5A for Brugada syndrome. And then, of course, a record about BCR-ABL1 fusions as a categorical concept with secondary resistance mutations that confer imatinib resistance in a particular cancer type. So all of this complexity, this is just, like a few of a wide array of complex and diverse types of knowledge statements needs to all be supported under a consistent framework that captures all of this complexity and all these various different use cases. So to do this, we operate across three different kind of large scale domains in GKS. There's our variant representation domain, our variant annotation domain, and our sequence annotation domain. And we're going to talk about all of these things over the course of our open house series. Today, of course, we're going to be talking a little bit about one of our products, the variant representation specification. We'll get to that in a moment. But just to give you kind of a high level overview of all the different products that we develop and use in our workstream, We've had two different study groups in GKS. The first is on categorical variation, the second on sequence annotation. The latter, sequence annotation, is planned for release in quarter 2 of this year. And then we have three actively developing products. These are actual specifications that are being designed for use in real world data systems. The first of these is Verse. Verse 1.3 has been active for a few years, but version 2, as Beatrice mentioned, is currently up for consideration by the GA4GH Steering Committee, and that vote occurs on January 13th. So this is version 2 is imminent. Now, VA Spec and Catverse are two upcoming products. We've had a series of different pre-releases that have been used and trialed out by many of our implementers, but these are currently planned for their initial version 1.0 release in the first half of this year. And we're very excited to see those kind of getting close to the finish line. And these pre-release versions and the released version of Verse have been implemented in a variety of different systems, and we have several upcoming works as well. Here's an example, or here's kind of just a high-level overview of a lot of different current and ongoing implementations in our community. During our open house series, we're going to invite people from each of these implementations to come and share their use of the GKS products in these implementations, so there's a firmer understanding about how these are helping them overcome their own goals for genomic knowledge and evidence sharing. And today, of course, we'll be hearing from the BRCA Exchange at the end of this call, or hearing from Melissa about the BRCA Exchange at the end of this call to see how they've used Verse, for example. Now, one thing I'd like to mention is that in GKS, we recognize that we are building new standards in a space where there aren't really any existing standards. And what this means is that we need to be aware of community needs and community risk in adopting these standards as we're developing them. And so we think about things in terms of the adoption life cycle, and we of course in the work stream tend to be all on the kind of innovator end of this spectrum. We're all people that have a driving need to see these specifications develop so that we can implement them and use that to share the data. But now, as we are moving into our initial or second releases of these specs, we're kind of moving into the early adoption phase of this chart, and for us to be able to indicate to the community which of these products are ready for, kind of broader uptake and testing and use, we've developed a maturity model. And this maturity model is used to help indicate for any of our products kind of where things are at and how ready it is for you to pick it up and start playing with it. Now, we have several different levels that we have planned for use. So that's draft, trial use, normative, and deprecated. And the idea here is that as we move from draft to trial use to normative, the product gets increasingly stable and increasingly ready for community use and adoption. Anything in normative, for example, you can expect, rock solid stability. Nothing in that model is going to be changing, and you can use that for the next 5 to 10 years and have no concerns about it. But anything at the trial use level, it has seen community testing. It's likely to undergo only slight changes, if any, down the road. And so, you can have a little more confidence picking that up and starting to use it even as an early adopter. And then stuff in draft, well, that's ready for those of you that are really like interested in, okay, I've got this, I need a model to describe this particular data type. And, I recognize that there's something out there that I could start trying out, but also have a lot of influence in shaping. And we do have some draft models that exist in our GKS specs today. Okay, so this is kind of our overview of how we, communicate to the community where the different specs are at, where the different features within those specs are at. And in order for us to advance in the maturity, we actually work very closely with all of our registered implementation projects. So here's a list of some of our implementation projects. These are all active contributors. And I know several of the members of these, several of the leads of these implementations are on today's call. And we'd certainly welcome any of your feedback on this process during our discussion section today. But one of the things that we strive to do is work with these implementations to say, okay, we're developing a new model for trial use. We think that we've got this nailed down. We've had a few successes with it in some of our driver projects. We want to make sure this works for the broader community. Please provide your formal feedback. And then they do. They will fill out a survey, they'll test it out, they get a review period, and then we use this in order to consider whether or not things are ready to be driven. So for example, for our Verse 2.0 work, one of the things we did was work with this implementer community, everyone who said, I'm going to be using Verse 2, to say, all right, which features are you planning on using? Which ones are you not planning on using? And we get this feedback, and it helps us decide what are the things that really are ready for trial use, and what are the things where we might need to take a step back and say this is still in draft. It really hasn't had broad community review or broad community adoption yet. And so we use this to help drive the development of our products. And of course, one of the things we want to do with our GKS Open House is welcome more product implementations into this process, people that want to use the GKS specs, that want to get engaged and want to have a role in the development of these. It's quite easy to do. We have a formal process for it. And if you're interested, we hope that you do get engaged. So okay, that's a high-level overview of who we are and what we do. As I mentioned at the beginning of today's call, this is our very first GKS Open House. We're going to be holding a series of these throughout the year. Our goal is to share live implementations and high-level product progress in each of these calls, which we're holding every 2 months at alternating times. And we'll record these and share these publicly so that people can review asynchronously if you can't make all of them. But we do encourage people to come and join us live so that you can participate in the corresponding discussions. And during today's call, we're going to be summarizing the GA4GH Verse product, as well as its implementation in the BRCA exchange. So I think that's it for the introduction. If there are any comments or questions, we'll have a short break for these after each of our speakers today. So I'd encourage if anyone has any high-level overview questions that they get them out now so we could talk through them. And then after that, we'll move into Verse 2.0, which Larry will walk us through. All right, very good. I'm moving on. So, all right, it's my pleasure then to introduce Larry Babb. Larry has been working on the variation representation specification, or VERSE, since the beginning of time. He's been with this project longer than anyone else I know, and is really one of the leading authorities on it. So please join me in welcoming Larry to walk us through Verse. Larry, just let me know as you want me to advance slides.
Right. Thank you, Alex. Excuse me, I have a little bit of a cold, so hopefully you'll be able to hear me fine. As Alex said, yeah, I've been working on this for a long time. The origination of this project was even before Global Alliance was formed. It was called the Variant Modeling Collaboration, and it was a group of individuals with Reese Hart and Bob Freemuth and some others that got this thing going. And eventually we merged into what is now Verse. We've been through Verse 1.0 and now we're up to Verse 2.0. So my job is to try to stay high level. Very hard for me, but I'm going to do it and go through this. So next slide, let's do it. For those of you that want to see the current spec, the best thing to do is just to go right out to the link at the bottom of the screen there, verse.ga4gh.org, and you will see a read the docs specification. When you go there, you'll see the stable one, which happens to be version 1.3, which is not exactly what you're seeing here. You're seeing the 200 ballot 2024-11, which is the one we're trying to get through product approval on in a couple days. And I thought it was just more apropos to show you that. If you do want to see this one and you go to read the docs at the verse.ga4gh.org and you say, how do I get there? Read the docs moved the way to get to change versions of the spec down to the lower right-hand corner in that little black box. So please go down there, click on it, go to that 200 ballot one. I just think it's the right way to look at it at this point because we are moving forward with 2.0. Okay, so what is Verse? Let me just give you the high level here. It aims to improve the computational exchange of variation from simple to highly complex forms. We assembled the, as you could hear from Alex's, driver projects and individuals to establish this and begin extending and maturing it with tooling and system implementations a while ago to realize these aims. Now, one of the things, it's really hard to describe, like, why do we create VERSE? Well, it's pretty hard to describe why did we create VERSE. There's all kinds of standards out there, and we're going to get into that. But essentially, it's really, we're trying to reduce the complexity and the chaos that goes on from the time variant calls are made after. Basically, once you make variant calls in whatever process you have, and you need to then go out and compare those variants to other patients' variants or other knowledge bases to get variant annotations, you know, everyone really is doing it their own way. And you can say, well, I'm not doing it my own way. I see how they're doing it and I'm doing it their way. But we're all implementing it, and there's no like super serious standard or rule around it. So we're trying to figure out how to change that so it can be done a little more computationally, reliably, and we can really get past this issue. So there's a number of scenarios. We can't get into them all here, but I'm going to give you a couple. If you're in a testing pipeline, from a testing pipeline perspective, and you're starting to filter your VCF results, one of the things we all do is we go through our whole genome sequences, whole exome sequence VCF files, and we annotate them with ClinVar, Civic, Nomad, and all that kind of thing. You can do that with VERSE now. You can versify your VCFs, and you can go against versified Civic and Nomad and all that, and just compare identifiers instead of having to do all your own matching to get that done. And you might say, well, that's pretty trivial. Everyone knows how to do that. There's toolkits, libraries, and all that. But it goes on. What if you want to compare, genomes to genomes, a variant in one genome to another one's genome, and see if someone in your genomic data hub has it? Well, to do that, you'd have to make sure you're all in the same reference build and all that kind of thing. With computed identifiers, this starts to become a lot easier in these databases. And so we're exploring that. And a lot of our implementers are out there doing this right now. They're working on these things. So let's go to the next slide and let's get into the details a little more. So here's an example of what's a very common pipeline of genotyping here, where you're just going through and you're using sequencing assay technologies, and then you're using a specific variant calling methodology to get your SNVs and indels for short sequencing reads. This is just one way to get VCF variant calls, and you'll end up with a bunch of variants. But it's not nearly exhaustive, obviously. There's lots of ways. It could be long read sequencing that you're doing here to get more than just SNPs and indels. If you go to the next slide, you'll see that there's a whole bunch of different ways in which folks go through and they apply different assay technologies for different kinds of biomarkers to get variant calls. And, this is not exhaustive either. Thanks to Alex Wagner for putting this together. I'm more of a software engineer, so I'm sorry if I'm fumbling a little bit with some of the scientific terminology behind this. But in essence, what we're trying to show you here is that this is where variants come from. These are observed variant calls at the bottom of the stack here. And this is where VERSE picks up. So let's go to the next slide. Once you have these observed variant calls from these different approaches, generally the next step for a lot of folks is I want to go out and look across all these variant resources to annotate this with, whether it be Splice AI or dbSNP or dbVar or Cosmic or ClinVar data or Nomad cohort allele frequencies or whatever it might be. But each of these different variant resources may have different ways of representing their variants inside their databases. So right away, the pipeline folks that are in charge of annotating pipelines have to start dealing with this. And as I said earlier, that's one of many things that we need to do. This doesn't even get into the human effort of how do you look these things up, but just computationally, this becomes challenging. There's a lot of these databases out there. There's more and more. There's also databases out there in hospitals and research labs and clinics and whatnot where they store these patient files. And wouldn't it be nice to be able to go in there and say, do you have any patients with this variant, and have them easily be able to look it up. And this is one of the things we're trying to accomplish with verse, or at least make it easier. I know a lot of people are trying to accomplish that. All right, so let's go to the next slide. So why do we need another standard here? You know, and again, I'm hopefully trying to delineate what the difference is. But you know, you see this XKCD comic that you've probably seen a million times out there, that all we're going to do is end up creating a 15th competing standard. And, you know, I almost took this slide out. We've been showing this slide for a while. I don't really like to consider verse as competing, you know, it's not a competition, we win. No, it's not. I'm not speaking like that. I'm basically saying that the verse here really is a complement to these other variation, quote unquote, standards that are out there. But let's talk about some of the use cases. And these use cases are very specific, in my opinion. Reasons why you would use VCF. Obviously, you would use VCF for compactness and computability. VCF is not going away anytime soon, nor should it. It's incredibly useful for what it does, and it will be around for a long time. But the issues with VCF are you're really locked down to making sure you have a common reference on a common genomic reference, I mean, for your variant calls when you're using VCF files. And you want to exchange variants in a flat file format. Now, a lot of folks will take this VCF format like Nomad does and create a kind of nomenclature from that, you know, using the chromosome dash position dash ref dash alt kind of thing to give it an identifier using a VCF style kind of name to it. And you'll see that a lot in a lot of databases. Let's go on to the next one. HGVS is much more useful for human readability. And while HGVS can do pretty much all the genomic stuff that VCF can, very few humans, I would think, use HGVS when they're referring to genomic coordinates because it gets a little cumbersome with large positional numbers. So it's generally used for things like DNA, RNA, and protein variants, and it's very useful for this, and it's not going away anytime soon. It also has a lot of differences in it that we can go into. You know, HGVS right shifts things when there's repeating variants in a sequence where one's deleted or inserted, and VCF left shifts things. But anyway, HGVS is very useful. Again, like I said, it's not going anywhere. It's littered throughout the literature, and rightfully so. It's really easy for humans to use and pass around. Okay, what's next? Oh, let's go to the last one. And verse. Verse is really not either of the two above. It's really more for decentralizing interoperability. It's more to appear in complex data contexts, and I would say it's much more of a computational use and less of a human use. And it's also used to represent variants on diverse sequences, a diverse set of sequences, meaning you can do it for any kind of sequence. All right, let's go on. So the variation representation specification was intentionally designed at its core to have a terminology and information model at the core of it. And that's really what verse is. It's the spec. And it's meant to be that in itself. Let's go to the next bullet there. It's meant to be interoperable across all formats. These are the symbol. That's a symbol for JSON. I just realized that this morning. Alex had to tell me that. Protobuf, XML, but any format you want to create a reference implementation for, you can. You'll see in the circle to the left, the top half of it with the little global alliance icons in it show you what we've been working on in the verse specification. So it's the read the docs specification that you're seeing there at the core. At the top in the light blue is a reference implementation we've done in Python called Verse Python. It's not, you know, it's the only one I know of out there, but anyone wants to go out and write another one in another language, they're more than welcome to do that. There's also a JSON schema protocol that we've created. We went with JSON schema out of the box because it just seemed to be the most useful and the most versatile, at least across all the initial implementers. And there's some GA4GH conventions that we've incorporated in there. We do not make policies about how you should call variants or what things are wrong or what things are right. We let you use verse and apply it to whatever the caller has called, or whatever a caller or a knowledge expert wants to represent as a variant. We don't judge it, we just allow you to represent it. Okay, next. Verse is also meant to be self-descriptive. So you basically, it contains everything that's needed to describe the variant. We often call these objects in verse value objects, in that the object itself is the identifier. It has exactly what is needed to be 100% identifiable and unique. Next. And finally, we want it to be globally consistent and decentralized. Again, getting back to this notion, and we're going to get into this a little more in a second, this notion of computed identifiers, being able to generate a unique identifier that anybody can do on their own local machine in isolation, and be confident that the variant that they're representing, if someone else has that same variant and they apply the spec consistently, they'll also be able to produce that same identifier and share it. And they don't need some authority to mint an identifier, register it somewhere, and go to some system that everyone has to depend on. All right, next. Here's an example of the anatomy of a verse variant. Again, this is a high-level overview, so there's a whole bunch of different kinds, but this is an allele. And at the core of it, in purple, what you're seeing is the sequence reference. For all sequence variants, that's sort of the baseline. You have to have that number line, or that sequence reference or reference sequence that is the very thing that you use to compare to other variants. So you know you're talking apples to apples. Next. Outside of that is the sequence location in green here. The sequence location is based on a reference sequence, or what we call it a sequence reference, not to confuse it with reference sequence. But the sequence location takes a sequence reference, and it gives you the start and end position. One of the other things to note very quickly is for verse, we use a zero-based indexing schema. So whereas VCF uses one-based position indexing for the sequences, we use a zero-based interbase position. So the 0, 1, 2, 3 for the numbering in verse represents the spaces between the bases, starting with 0 before the very first base. Hopefully that made sense. Again, we can get into the details later. So this shows you that this is talking about the base between positions 7400 and 7401. And then finally, go next one. The allele itself out here is a combination of a location and a state. The location, like I just told you, is on that reference sequence between those two positions, between those two bases. Positions 7400 and 7401, that base will have the state of a C. And that's the little sequence expression that goes there. So this is what we call an allele. This is one of our very core baseline, and is used quite a bit. And this is one of the many things that we're working on trying to get productionized. And as you can see at the top, that gives you the sort of the HGVS representation of it in minimal form. Okay, next. So as I had mentioned briefly, and we don't have time to get into this, but there's a whole, in the specification, there's a whole protocol and methodology for how you compute the identifier. You take that minimal form that we saw on the previous page and take it through a certain amount of processes there. And basically what it allows you to do is digest it down into what's called a GA4GH computed identifier, which you're seeing there in green. And it starts with the namespace GA4GH colon, and the VA that you're seeing there stands for variant allele, VA, and dot, and then you'll see a UUID like kind of thing afterwards. That's basically the digest, and that digest is guaranteed to be unique if you have the same state at the same location on the same sequence. So those three representations you see up there on the left are basically the same exact reference sequence, even though they're given different accessions by different groups. And since they're the same exact locations and the same deletion, then they will end up generating the same identifier. So you can see where the advantages start coming into play here is that when you want to now search, imagine if folks created resource indexes based on computed identifiers for the HGVS expressions and VCF and nomad expressions and whatever expressions for the variants in their publications or in their database. Now, all of a sudden, you have a much simpler way of finding all the references, not just the one that matches your HGVS expression. By the way, this computed identifier concept was born out of some work at Invitae by Reese Hart and some others. I don't know. I'm sure there was others at Invitae that did this. And this is where it really sort of evolved into. They used it quite effectively for many years in all their pipelines up until, and probably now as their lab core in their pipeline processing. Okay, next. The current status is of VERSE is, and we can go through this fairly quickly. We produced, we put out several publications over the last few years, starting with Cell Genomics publication in 2021. That was sort of our marker paper there for when we first got things off the ground. And we subsequently have had two other papers go out. One on genotyping, on how we're going to represent genotypings and pharmacogenomics. That's to come yet. It's not in verse 2.0 officially, but it's something we plan on work on in the near future, and we can discuss that if anyone wants to. And then there's some great work being done by Alan Rubin and their team at MAVE, the MAVE DB group, to take the custom sequences in these functional assays from different organisms and whatnot, map them to human organisms using computed identifiers, and put them into a nice database. And hopefully we're going to see that come out soon in a VA format with all the verse 2.0 stuff. We also have integration with phenopackets. That's already been integrated. Hopefully they're going to be looking at going to 2.0 soon. And we're working quite a bit with Beacon right now. Beacon seems very eager and interested to align with some of the stuff we're doing, because as they're, those of you that know what Beacon is, they're basically trying to create this federated network where people can reach out and say, does anyone on this network have this variant or have seen this variant? And even though that seems like a simple ask, it's not that easy. And they're finding that there's some value in the way we're doing things here at Verse and some of the tooling we've created that might make it easier for both the implementers to set up these Beacon nodes and be able to search by it. That's my opinion, and so we should wait and see how that develops. All right, next. So Verse 2.0, we want it to be a lingua franca for variation, and not just for small variation like I've been talking about here. But you can see there, there's a representation of how you might represent two alleles as a genotype. In the far left, that's a molecular kind of variation. And then there's structural variation, which is also molecular because you're really talking about the change to a structure of the molecule, even though it's something like a translocation ends up changing two molecules. And then finally, over to the right, we have copy number, which we refer to as systemic. To me, this is confusing because as I've been learning about copy numbers over the years, and I still don't have that group nailed down, there's copy numbers that are considered duplications within a molecule that they care about. And there's also copy numbers where they care systemically how many counts there are within the whole genome. And even though they call them both copy numbers, they are little different beasts. And that's my opinion on copy numbers. But anyway, we have a way of representing both, and hopefully this will help us distinguish when we're talking about one versus the other. All right, next. All right, in verse 2.0, one of the things we've done, if you look at that diagram to the right, the text to the right, the JSON there, that's basically the same JSON we were looking at before with a minimal representation. What we did in 2.0 is we decided, we need to be able to allow our developers, our engineers, our systems folks to be able to annotate and decorate the verse objects themselves so they can pass along all this useful information that they're so used to anyway. And it's so necessary for display and for human purposes and reporting and blah, blah, blah. So we allow you to put labels and different kinds of expressions, whether it's HGVS or nomad expressions, and you can put accessions on it. We have the alphabet for the reference sequence. Is this a nucleotide or is it a protein kind of alphabet kind of thing? And you can put the reference sequence in, not just the alternate sequence for the state, that kind of thing. And on and on and on. At first, in 1.0, and we did not allow this. We were keeping our verse objects to be purely just the data needed to identify it and create it, which was really nice and pure, but it was not realistic from an engineering standpoint. So this has really opened things up. We still generate the ID based on that minimal information. We do not include the boldface data that you're seeing here. We only include the unhighlighted stuff or non-bold stuff. All right, next. I'm going to pass it back to Alex because he's going to talk to you a little bit about what's coming up in verse 2.1.
Okay, so yeah, with verse 2.0 just around the corner, we're thinking about some of the things that are coming up in 2.1, and some of the things that we've discussed during, both 1.3 as well as 2.0 are some variant types that still need a little more work in order to be drafted and put into verse. And so this is a great opportunity for people to get involved. If you're interested in how we represent fusion data, how you represent phasing data and genotypes, these are some of the active topics that are coming up in verse over, say, the next 6 to 9 months. So if these sound like things that are relevant to your work, this is a great time to get engaged in the verse project. Okay. So I think that wraps up the verse presentation. We've kind of run through with the presentation and discussion time, but if there are any like high level pressing questions, maybe we've got time for like one question right now. So if you have one, please raise your hand using Zoom. Otherwise, you can hold on to it or write us later, and we're happy to get back to you.
I knew there wasn't going to be a question. That's why I took the whole time.
Oh, good. All right. Great thinking, Larry. Nicely done. Uh, Okay, well, very good. So yeah, if one occurs to you later, or if you're watching this asynchronously and you're like, wait, I want to know more about X, Y, or Z, please don't hesitate to reach out. We're more than happy to address your questions asynchronous. Okay, so with that, it's my privilege to introduce
Wait,
Dr.
wait.
Melissa
There was a
Klein.
question in the Slack, Alex, and you might be able to answer it super quick. I don't know. The example in C.7280 to C. I think that was but the start was for 7400. Yes. We're showing there the transcript label, right? But oh no, it's on the transcript. So do you have any thoughts on this? Why is it 7400?
This might just be a typo. I'm not
Okay.
sure, or maybe there's different transcripts. I'm not certain. We'd have to go back and look at this example, but very sharp eyes, Ali Shuang. Thank you
Yes.
for pointing that out. We'll definitely come back, and
Yeah,
if
we'll
if
clean that
there's a
up.
reason for that, we'll
All right, thank you.
label it. All right. Cool. So coming back to this, it's my privilege to introduce Dr. Melissa Klein. Dr. Klein's worked in GKS also since the beginning of time. I know she's been with them longer than I have, so it's a real privilege to hear about all of the experience that she has had that drove her work in GKS as one of the driver project champions for the BRCA exchange driver project. So please join me in welcoming our GKS co-lead, Melissa, as she talks to us about the BRCA exchange.
Thanks, Alex. And yeah, well, now you've heard what we're trying to do with GKS, what FIRST in particular is, and now I'm going to give you the user perspective applying this technology. So next slide. First, motivation is how genetics strongly influences the risk of breast cancer, as well as many other heritable disorders. And so people who undergo genetic testing these days often have the opportunity to better understand and clinically manage their heritable disease risk. In the case of breast cancer, there are ways that people can avoid getting cancer altogether if they know about the risk. But first, they've got to know about the risk. And next slide, please. And here is the problem, that for what turns out to be the majority of known genetic variants out there, we don't have the clear clinical annotation of which variants are presenting, disease risk and which aren't. So here we're looking at BRCA1 and 2, which are better studied than the genetic variants in most genes out there. Looking at what's in ClinVar, we see some pathogenic, some benign variants. We see that almost half of the variants that have been reported to ClinVar are either of uncertain significance or there are conflicting interpretations, meaning that the clinical knowledge on those variants is still muddy. But this is dwarfed by the number of variants out there just in Nomad that haven't even reached ClinVar yet. And um, and anything in Nomad, it's not a hypothetical variant. These are genetic variants that have been observed in recent times in sequencing studies in people who are walking this planet. So this gives us a glimpse of the annotation bottleneck that we're looking at. And next slide, please. And so a question, how do we classify variants? Well, by the ACMG guidelines, we take a number of different evidence types together, integrate all of the data that we get, and then the ACMG guidelines and the VSEP rules, if there is a VSEP, a variant classification expert panel, they define a system for weighing together all of this evidence and assessing whether in aggregate the variant looks pathogenic, benign, or somewhere in between where we can't yet say. Next slide. But the little piece that's key, and that's the stumbling point actually for most variant interpretation communities, is pulling all of this information together. So years ago, the GA4GH launched what became the BRCA Exchange to assemble a one-stop shop for BRCA1 and 2 variants, and as an exemplar for other genes and heritable disorders. And so we pull together data from public repositories and publications selected by the expert curators, annotation pipelines, and there's the URL. Next slide. And so here's what it all looks like. For a given variant, we present a number of different identification terms, clinical assertions, existing curations of the variant as benign, pathogenic, something in between, and some of the evidence that can be used to interpret the variant. Next slide. And now we're getting into how do we use GKS? First, the first step is we support verse digests. Among the nomenclature terms that people can see on the screen and can search for programmatically, we include the verse identifiers. Maybe somebody somewhere has been copying and pasting these identifiers to do a variant search on other resources, but the much larger use case is that people who are accessing our data via API can query based on this digest and can, when they retrieve the variant data, they'll also retrieve the digest. So this enables the support to see if this variant is the same as some additional variant in your own repo. Next slide. And so next part is, well, we pull together variants from multiple different sources, and it's important to pull together all of these different sources because each different source presents different variants and also a different slice of knowledge on those variants. Next slide. And so, putting that all together in our backend pipeline, we first take all of the different variant sources, translate them to VCF on the HG38 reference sequence. There are major chunks of the pipeline that you're not seeing in this diagram to do that. And then merge all that data to generate a set of distinct variants and merged information which goes on bracket exchange. Now, traditionally, well, in the beginning, we did this we compared these variants by generating the alt alleles of the variant on HGVS, the variant on HG38 with some flanking bases, and did a string comparison to see which variants were actually the same. Now we use now we're using the Verse Python VCF annotator tool, which slips neatly into our workflow, and that computes the verse digest for each variant so that we can just merge the variant data based on those digests. And it's similar conceptually to what we were doing before, more robust. And the great advantage is that this extends naturally to to structural variants and copy number variants and other types of variants that we hadn't been able to support in our previous pipeline. I wanted to say a little bit about the implementation experience. So generating the Verse Python digests in bracket exchange. So installing, well, Verse Python is open source. Installing the dependencies takes, I would say, medium Unix skills. Once you've got the dependencies installed, installing Verse Python itself is easy. It's a simple matter of pip install and off you go. There are lots of examples in the code base. Running the VCF annotator is slightly more involved. You've got to install the GitHub library itself, but it's not bad. Next slide. Moving forward, as we're looking at better ways to package the data, particularly as the variant annotation specification rolls out. When we look at each tile or each nugget of information on bracket exchange, each of them is a type of annotation, and the VI specification is a structured way of presenting the annotation with its underlying evidence and provenance data. and so we are looking forward to, to packaging up our data for download and for API according to the VA spec framework that will both improve the modularity of our data via download, API, what have you, and it will also present the data in a structured form, which will hopefully become more familiar as the standard gains uptake. And next slide. So we wanted to leave some time for questions and for discussion, questions and comments, but first off, thank you for your time this morning. And if you've got any interest, get involved. You can contact Beatrice as a starting point, whether you're interested in getting involved in product development or getting involved as an implementer, test driving one of these products in your own code base. And so at this point, next slide, I want to turn it over to any questions or comments you might have. And thank you all for your time this morning.
Okay, well, we'll wait just another moment for any last questions. We do have 3 minutes left for the end of the hour here. I will say that there was an earlier question about the C.7280T to C. Corey did provide an explanation for the difference in that. It has to do with where the CDS is indicated in the reference transcript and how HGVS nomenclature starts from the CDS, whereas verse starts from the beginning of the entire transcript sequence. If you've got more questions about that, do feel free to reach out or follow up. Otherwise, I would just say that it's been a real joy to share all of our progress on Verse and to hear about this implementation work at the BRCA Exchange from Melissa. Um, if there are any, if you have any follow-up interest in this, of course, Beatrice is the best first point of contact for the work stream. But if you're watching this asynchronously and you're like, oh, I really wish I was in that session and could have asked a question of Larry or of Melissa or of Alex, please don't hesitate to write us directly after the fact. We're more than happy to field any follow-up questions you might have. Okay, so if there aren't any follow-up questions from today's presentation from those of you here today, I'd just like to join in on Melissa's comments and thank you all for your time and attention at our first GKS Open House. Beatrice, I'm going to put you on the spot. I'm terribly sorry. When is our next open house? And while you're, sorry, go ahead.
No, I was about to say that will be in 2 months, and I'm happy to share the link to the calendar invite if you haven't got that already. That will be accommodating the Australian Pacific region, so that'll be at a slightly later time. Yeah. Were you just about to finish with something?
Oh, no, that's it. That's great. Thank you.
Okay, well, thank you everybody for joining us, and thank you to our speakers for a wonderful session. Really appreciated that. I've written a couple of notes in the chat, basically the recording and the slides will be shared afterwards. If you'd like to join, please do let me know, and I'm happy to share link to the meeting notes with you. If you have further questions, as Alex, Melissa, and Larry just mentioned, do please let us know. But thanks again, and do hope to see you at our future events. Bye-bye.